Hello, my name is Stacey Ward, and I am an R&D manager within Cell Design Studio in MilliporeSigma. And today, I will be presenting on our genetically engineered immuno-oncology cell lines and services for early drug discovery research and preclinical manufacturing. Within the U.S., the life science business of Merck KGaA operates as MilliporeSigma. For the talk today, I will begin with an introduction to our Cell Design Studio, as well as some immuno-oncology challenges that we have identified within the field. I will then move on to a discussion of our antigen panels, and finish up with a discussion of our monoallelic HLA panel. The Cell Design Studio is a custom cell line engineering service group within the life science group of MilliporeSigma. We use gene editing tools, including CRISPR-Cas9, zinc finger nucleases, short hairpin and siRNA, and lentiviral overexpression to engineer custom cell lines for our customers. We use many different cell types, including pluripotent stem cells, primary immune cells, and transformed cell lines. And we can pretty much engineer any type of modification that you might desire, including single nucleotide polymorphisms, gene knockouts, large reporter cell lines, and such. We use many screening tools, including next-generation sequencing, digital droplet PCR, and junction PCR for genomic interrogation, as well as phenotypic assays, including flow cytometry, microscopy, as well as things like cell migration in a dish. And the cells that we engineer can be used for any downstream application, including in vivo testing, IPS differentiation, and high-throughput screening, to name a few. But at the end of the day, what we do is we genetically engineer cells that are modified to our customer specifications. And while doing our cell line engineering service for our customers, we have started moving into the immuno-oncology space. And what we found within the field is that it is a very long and arduous process to get from identifying a disease that you might be interested in attempting to treat and getting to the clinical phase of actually testing your treatment in people. So we wanted to use our skill set at engineering cell lines to try to both improve the cost and shorten the timeline for our customers. And when we look at this timeline, once a disease has been selected to potentially target, a target ID needs to be identified and/or a pathway. Many assays need to be developed in order to test these targets or pathways in vitro, and most often or not, these include some sort of high-throughput screening assay. And then once a customer has a hit that they are interested in pursuing, there is also a space for us within the lot release and preclinical testing of whatever pharmaceutical or target that they are interested in identifying. So where we thought that we could fit in is within the assay development phase of this, where we can engineer cell lines that our customers can use to screen for their targets or for any sort of lot release assay. And when we think about what these targets could potentially be, this is a screenshot from a figure from a paper published in 2019 that shows the target landscape compared for drugs in the pipeline in 2017 to 2019, so only 2 years difference. What we can see here is obviously there is a wide increase in the number of targets that are going to be investigated within just those 2 years. In addition to that, the size of each of these bubbles is directly related to how many potential drugs are targeting each of these targets. Boxed in red on the right are 3 such targets, including CD19, EGFR, and NY-ESO-1, which are 3 of the targets that we decided to pursue for some of our off-the-shelf cell lines that could potentially be used for early drug discovery as well as preclinical testing. So while working with our customers to engineer their cell lines, we have identified some challenges within the field. These start with the development pipeline, including target identification from patient samples. So once a disease has been identified as a customer being interested in potentially targeting, these patient samples prove very valuable to try to identify either mutated genes or pathways that could potentially be a target. However, the utilization of transformed cell lines for clinical pre-discovery and compound and reagent testing are also equally important, since patient samples do not typically grow very well in culture. So the current limitations within this pipeline include things like in vitro testing that requires negative control cells that do not express the target of interest, and positive control cells which do express the target of interest. And what we have found is that currently used control cells may not be from the same person, organ, or even lineage, which can lead to high background and false positive and negative results. So we have identified that the field needs non-passage-limited, easy-to-grow, and scalable cell lines that are pertinent to clinical endpoints. If these cells are amenable to 96 384 well assays, have affordable and easy culture conditions, and are able to produce robust and reproducible results. And this is where we thought that our strengths in cell line engineering could come into play. This is an example of some of the target cell lines that are typically used in drug discovery and preclinical testing. This is a paper from 2013, which was looking at two B-cell lymphoma targets, CD19 and CD22. Positive and negative controls were required for these assays, so a cell line that expresses CD19 but not CD22, or CD22 and not CD19. And what this group did was they simply took the available cell lines that are easy to culture, and they assayed them for how much protein they expressed on the surface, as shown in this panel on the upper left-hand side. And what you can see on the bottom, part B, is that these cell lines do express varying levels of each of these proteins. But when we look at the primary patient samples on the right-hand side of the slide, they also express varying levels of these proteins. You can easily see that this is a very complicated set of cells to attempt to use for any sort of preclinical testing or lot release, since they express varying different levels of the proteins. So we asked the question of, what if instead of using a mixture of cell types from different organs or tissues or lineages, you could use a matched panel of cells that grew with the same media and similar growth kinetics and morphology for your preclinical studies? And we set to do this for what we're calling our tumor-associated antigen panel cell lines. We have identified parental cancer cell lines that grow very well in culture and typically express the tumor-associated antigen of interest endogenously. We performed genetic knockout experiments using zinc finger nucleases to render these cells null for this tumor-associated antigen of interest, thus giving you a negative control cell line. We then took these knockout cells and we delivered the tumor-associated antigen of interest via lentivirus, and we performed single-cell cloning and identified clones that expressed differential levels of the protein, thus giving you one cell line that expresses low levels of the protein and another cell line that expresses high levels of the protein. But all of these came from the parental cell line. They grow the same in culture, use the same media conditions, and they are amenable to high throughput screens. The three cell lines that we chose to use for our three tumor-associated antigen panels that I will be presenting today include Raji cells, which are a B-cell lymphoma cell line that express the CD19 antigen of interest, A549 cells, which are lung cancer cell lines, which express EGFR, and A375 cells, which express the NY-ESO-1 peptide. This is the data that we generated on our Raji CD19 antigen panel. CD19 is a cell surface molecule that is expressed on the surface of B cells and highly expressed on the surface of B-cell lymphomas. We generated our knockouts using ZFNs and used next-generation sequencing to confirm the genotype of these cells, as well as extensive phenotyping using flow cytometry. On the right side of this slide, you can see that our knockout cell line does not express CD19 on the surface, and we were able to find two single-cell clones that expressed either a low level of CD19, shown in red, or a high level of CD19, shown in blue. And I would like to point out that these low and high phrases that I am using are in general relative to the endogenous level of the protein on the parental cell line before we engineer the knockout. For our EGFR panel, we chose A549 lung cancer cells. EGFR is a receptor tyrosine kinase that is known to be mutated in many different types of cancer, including lung cancers. We, again, used ZFNs to generate knockouts. In A549 cells, there are actually 3 copies of EGFR within the genome, so we were able to knock out all 3 copies within our parental knockout cell line. And then when we redelivered the EGFR expression using lentivirus, we were again, shown on the right, able to find a low expressor and a high expressor. In addition to delivering wild-type EGFR into this panel, we also delivered a commonly found mutant of EGFR. So EGFR V3 is a naturally occurring variant of EGFR, often found in cancers. This is an in-frame deletion, which actually causes EGFR to lose its ligand dependence and become constitutively active. So it is always signaling when you have this EGFR V3 mutation. We took the same EGFR parental knockout cell line that I showed you on the previous slide, and this time we delivered the EGFR V3 variant via lentivirus. And what was interesting is that we were unable to find a low or a high expressing clone. All of the clones that we assayed had the same level of EGFR on the surface. And when we think about this biologically, it makes a lot of sense because this is a receptor tyrosine kinase that performs intracellular signaling upon binding with a ligand, but since this variant 3 loses its ligand dependence, it's always on. So you can think of it as EGFR typically works as a dimmer switch, so either it's off in the absence of ligand. If you have a little bit of ligand, it might be a dim signaling pathway, but if you have a lot of ligand, it's going to be signaling quite a bit. With the V3, it's more like an on-off. Either you have the V3 and you have signaling, or you don't express V3 and you don't see any signaling. And that data is shown on the right-hand side of the slide, where we were only able to find 1 level of EGFR V3 expression. However, when we used a pan-EGFR antibody, we do see quite high expression of this protein on the cell surface. The third tumor-associated antigen panel that I would like to talk about today is a little bit different. So NY-ESO-1 is not actually a protein-coding gene, it is a peptide. So it is encoded by 2 genes, CTAG1 and CTAG2, which are present on the X chromosome. The NY-ESO-1 is a derivative of the protein product of these 2 genes, but it is a small peptide that has been found to be presented on the surface of many different cancer cells. And this includes colon cancer, and A375 cells are a commonly used colon cancer-derived cell line. So this was a little more complicated since the NY-ESO peptide is potentially encoded by 2 different genes. But again, using our ZFN system, we were able to cleanly find clones that had a knockout of all of the CTAG1 and 2 genes, showed in the middle panel using our next generation sequencing pathway. In addition, the NY-ESO is not, as I said, it's not a full protein, it's more like a peptide. So instead of using a flow cytometry phenotyping assay, we decided to use a Western blotting assay so that we could see all of the peptide that was potentially expressed within the cell. And you can clearly see that our knockout cell line in the first lane does not express any CTAG gene product. And in this case, we were able to find both low expressing clones and high expressing clones. So to summarize, I have shown you data on our antigen panels that include cancer-relevant cell types that express varying levels of the antigen of interest. And we feel that these tools will be useful for early drug discovery, antibody compound testing, or assay development. And due to our experience in custom cell line engineering, if this is something that is of interest to you, we can also develop custom antigen panels in your cell line of choice. So whether you have a particular cell line that you work in or a target of interest that we have not covered in today's talk, please reach out to us and we can generate a custom cell line for your uses. So to change gears a little bit, the next panel that I would like to talk about is our HLA panel. So far I've presented data on tumor-associated antigen expressing cells, but something that we need to remember within the immuno-oncology field is that it's not as simple as a cell line just expressing a target of interest. So when we think about the immune system targeting cancer cell lines or tumor cells, we need to also think about specifically the T cells. So I really like this cartoon on the left-hand side of this slide, where you have a somatic cell that is expressing a protein or a peptide of interest within an MHC Class I, and a cytotoxic T cell comes along and says, I'm sorry, but I don't recognize the MHC peptide complex you have there. And it makes him sad, because now the T cell does not recognize him, can't kill him, can't do anything, and just goes along his way. So what we need to remember here is that T cells will only be stimulated to target tumor cells when the TCR recognizes an MHC-bound peptide. And these HLA-dependent therapies can include cytotoxic T lymphocytes, as well as peptide, RNA, or DNA vaccines. And to look at this a little bit more in detail, this is a cartoon that I'm using from a paper published in 2008, which shows on the bottom an antigen-presenting cell, and on the top an MHC HLA-B8 restricted T cell. So the T cells will only recognize peptides as presented by HLA-B8. On the left-hand side of this slide, this is a T cell that can recognize this peptide in the HLA-B8 group. However, in the middle, we have the T cell and we have the correct peptide, but the antigen-presenting cell, instead of putting it in HLA-B8, it is putting it in HLA-B44. So even though it's exactly the same peptide, the T cell does not recognize it because it doesn't see that MHC type 1. On the right-hand side, the antigen-presenting cell is expressing the correct HLA, but it is actually presenting a different peptide. So that also is not recognized by the T cell. So what we can think of with this system is a very discrete lock and key mechanism, where the T cell you can think of as the lock and the antigen-presenting cell as the key, and everything needs to completely match up for that T cell to recognize the antigen-presenting cell and potentially kill it, which is what we're looking for with immunotherapy. So again, using our expertise in gene engineering, we thought, well, this makes for a very complicated potential in vitro testing system. When you think about MHC Class Is, you receive HLAs A, B, and C, one copy from each of your parents, so that's potentially 6 different HLAs that you are presenting on the surface. And while that's really great for the biology of your immune system, because it gives your T cells a lot of diversity, it can make assays with these cells quite difficult, and the data can be a little bit messy. So using a protocol that was first published in 2015, we thought, okay, well, what if we can knock out all of the endogenous MHC Class Is on our cell line and only produce one? And that way you're not limited by the cancer cell line anymore. If the cancer cell line of interest that you're using only expresses HLA-A1, but the T cell that you're interested in testing expresses or recognizes HLA-A2, you can no longer use that cancer cell line. But what if we can take that cell line that you're used to culturing and you're very comfortable with, and we can just engineer it to express HLA-A2? Then you have a system that you can use in your lab. So instead of the laborious task of attempting to knock out 6 different genes, all of which differ by only a grouping of single nucleotide polymorphisms, we decided to knock out a partner protein known as beta-2 microglobulin. This is shown in the diagram on the lower left-hand side. Beta-2M is an important part of MHC Class Is because it is what chaperones the MHC Class I complex to the cell surface. So without beta-2M, even though you still produce HLA protein, it cannot get to the surface. And when we think about T cells interacting with tumor cells, if it's not on the surface, the T cell can't see it. So we used DFN to knock out the beta-2M gene, and we carefully confirmed that these were true knockouts. So we did this using sequencing as well as phenotyping assays just to make sure that we weren't having breakthrough or erroneous expression of the HLAs on the surface. And we have not seen that in any of the cells that we've tested. So once we have this beta-2M null cell line, we again use lentivirus to deliver single HLAs into the cell, and we confirmed the expression via phenotyping. So we chose 3 different cell lines. We used A549 cells, again, our lung cancer cell line, the Jurkat cell line, which is a T lymphocyte cell line, and Raji cells, which are a B lymphocyte cell line. And on the right-hand side of the slide, we picked some of the most commonly found human MHC Class I, shown on the right. We focused on HLA-A and HLA-B for this first panel, and we expressed each of these HLAs singly within the cells. In this slide is a representation of some of the data that we generated using these panels. So for this example, I am showing data from the A549 HLA panel. On the upper right-hand corner, wild-type A549 cells are shown here in blue, expressing beta-2M on the surface, and our knockout cell lines are shown in red. So when we knock out beta-2M, you no longer get beta-2M on the surface. We have also stained these cells with pan MHC Class I antibodies, and similar to the beta-2M expression, we also do not see any expression of HLAs on the surface in our knockout cell line. Once we delivered each of the single HLAs shown here into this knockout parental cell line, you can see very clearly that we get high expression of beta-2M on the surface. And again, when we stain these cells with a pan MHC Class I antibody, we see similar results. So we are getting high expression of not only the beta-2M, but also the HLA on the surface. And we have also done internal testing looking for whether any of the endogenous HLAs are pulled to the surface once we overexpress these HLAs. However, with our lentiviral construction, we have fused beta-2M to each of these HLA types, so we only see the fusion protein product on the surface. We don't see any of the endogenous HLAs, but we have carefully looked for each of these panels. So to summarize the second part of the talk, we have developed cancer-relevant cell types that express individual MHC Class I alleles. And we have done this in A549 cells, Jurkat cells, and Raji cells. And I only show data for the A549s, but the data for the Jurkats and the Rajis look very similar. These cell-based assays can be used to evaluate TCR therapies, as well as peptide and oncolytic vaccine development. And just to point out, if you have other HLAs that you are interested in expressing or another cell line, other than the 3 that I've talked about today, we can, of course, do this on a custom basis. So I hope that I've convinced you that the utilization of one cell line that expresses varying levels of a tumor-associated antigen will aid in your preclinical discovery and validation, and that monoallelic HLA Class I expressing cells will be a useful tool for any immuno-oncologist interested in peptide presentation and T-cell killing assays. The data that I've shown today is all encompassed within our off-the-shelf tumor-associated antigen and HLA cell lines. You can find the website here. In addition, as I mentioned, we do provide custom cell line engineering as well, so please visit our Cell Define Studio webpage, and we would be happy to provide you with any additional information you may want. So just to finish up, using our expertise in tailoring cellular models to customer needs, we can perform any sort of modification that you might envision, including knockouts, target integration. We can deliver clones or pools. We are very experienced in induced pluripotent stem cells and primary stem cell culture. And any of these cell lines can be used for any number of drug discovery applications or other applications, even if you're not within the drug discovery field. And we really do look at ourselves as research partners. We are not a CRO, we are a partner with you. So you will have access to our PhD level scientists and over a decade of experience in engineering cells, and it's rare that we find a cell type that we have not engineered in the past. So I encourage you to please reach out to us with any questions you may have. So with that, I would like to thank you for your attention, and I hope that you have a wonderful day. Thank you.